Donc tout d'abord,
HRAC, which stands for Herbicide Resistance Action Committee, is the classification of herbicides that allows to quickly and easily know the mode of action of a herbicide molecule in the plant. There are currently 21 different groups. In brief, first, two molecules belonging to two different HRAC groups have two different modes of action. Second, all the molecules of a same HRAC group have the same mode of action and the same site of action. Let's see now the HRAC A group in detail. This group comprises 19 different molecules, which all have the same action in the plant. They inhibit the ACC-ACE enzyme essential to the plant growth. Group A includes 3 different chemical families, which means that they have different skeletons. There are 10 molecules belonging to the FOP group, 1 molecule belonging to the DEN group, and 8 molecules belonging to the DIM group, including Clethodim. This skeleton difference is essential. Although all these molecules bind to the same target, the ACC-ACE enzyme, they do not do it in the same way. This explains, for example, certain differences in terms of resistance patterns. Resistance is an inheritable characteristic, conferring on certain plants of a given adventive population the capacities to survive and to reproduce following an herbicide treatment, which, in normal conditions of application, would have controlled the said population as expected. Applications of herbicides therefore do not create the resistances, but select naturally occurring resistant individuals in the adventive populations. In fact, resistance to herbicides existed long before the introduction of herbicides. INRA researchers recently demonstrated from an herbarium collection of foxtail grasses harvested between 1788 and 1975 that one plant contained a mutation on the ACC-ACE gene, one of the causes of resistance to ACC-ACE inhibitors. This plant was harvested in the Bordeaux area in 1888, 90 years before the first ACC-ACE inhibitor was marketed in France. This mutation, located at position 1781, is now the most widespread one in Europe. For herbicidal efficacy to occur, the active substance must reach its target. Any mechanism avoiding this is a resistance mechanism. There are 2 major resistance mechanisms for ACC-ACE inhibitors: detoxification resistance and target site resistance. Detoxification resistance. The active substance does not reach the target. It is metabolized beforehand by the plant's enzymatic machinery. Target site resistance. The most known and most frequent mechanism is the modification of the target. The active substance migrates within the plant until it reaches its target, but it has a different structure that disturbs the key lock system. This difference is located on the gene coding for the enzyme, at the very heart of the DNA. Let's see what happens in the cell. When the mutation point at position 1781 is present, the difference is such that, in general, FOPs, DINs, or DIMs cannot effectively bind to the enzyme. However, contrary to other DIMs, Clethodim can effectively bind thanks to its different skeleton structure and can thus control the individuals carrying this mutation. Following various research work, at the INRA in particular, we have been able to determine the various mutation points on the ACC-ACE gene and their corresponding frequencies. Knowing that a plant is resistant to a particular molecule does not always make it possible to infer with certainty all possible cross-resistances. And, conversely, it is not because a mutation has been detected that all the molecules in the HRAC A group will be affected.